Hello, friends. Once again, good evening. We are again on Saturday with the reading of Biology of Plants, and we continue in the chapter number 3 related to the plant cell and the cell cycle. And we are going to continue with the parts of the cells, now the endoplasmic reticulum. So the idea, the let's say dynamic of this streaming is that I start reading, and once I have some question how to pronounce, then I check in the Google Translator and check how it's how it should be pronounced. And then I learn, and maybe you learn. That's the point. And in the middle, we review some concepts, important concepts about the plant. So for today, the plan is not being so long. So maybe with endoplasmic reticulum, paragraph, and Golgi apparatus. Uh, because the next cytoskeleton, no, this will be for the next week. So let's start. Endoplasmic reticulum. The endoplasmic reticulum is a complex three-dimensional membrane system that permeates the entire cytosol. In sectional view, the endoplasmic reticulum appears as two parallel membranes with a narrow space, or lumen, between them. The form and abundance of this system of membranes vary, vary is mucho, vary. Let's check this. Let's ask to the Google translator.
Vary,
Very,
vary,
very. It's quite similar. So very, greatly, 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 or greatly from cell to cell. So the form and abundance of this system of membranes vary greatly from cell to cell, depending on the cell type, its metabolic activity, and its stage of development. For example, cells that store proteins have abundant raw endoplasmic reticulum, which consists of flattened sacs, or cisternae, singular cisterna, with numerous polysomes on their outer surface. In contrast, cells that secreto. Let's check this. Secretar,
secrete,
secrete. Okay. So, in contrast, cells that secrete lipids have extensive systems of smooth endoplasmic reticulum, which lacks ribosomes and is largely tubular in form. The tubular form is involved in lipid synthesis. Both rough and smooth forms occur within the same cell and have numerous connections between them. These membranes are in continuous motion and are constantly changing their shape and distribution. In many cells, an extensive network of endoplasmic reticulum consisting of interconnected cisternae and tubulus is located just inside the plasma membrane in the peripheral or cortical cytoplasm. The most likely function of this cortical endoplasmic reticulum appears to be in regulating the level of calcium ions in the cytosol. The cortical endoplasmic reticulum may therefore play a role in a host of developmental and physiological processes involving calcium. It has also been suggested that his cortical network serves as a structural element that stabilizes or anchors the cytoskeleton of the cell. The cortical ER, the endoplasmic reticulum, let's say, is believed to be a general indicator of the metabolic and developmental status of a cell. Quiescent cells have less, and developing and physiologically active cells have more. Some electron micrographs saw the rough endoplasmic reticulum to be continuous with the outer membrane of the nuclear envelope. As we have mentioned, the nuclear envelope may be considered as a specialized, locally differentiated portion of the endoplasmic reticulum. When the nuclear envelope breaks into fragments during phase of nuclear division, it becomes indistinguishable from cisternae of the rough endoplasmic reticulum. When new nuclei are formed during telophase, vesicles of endoplasmic reticulum join to form the nuclear envelopes of the two daughter nuclei. The endoplasmic reticulum functions as a communication system within the cell, as a system for channeling materials such as proteins and lipids to different parts of the cell. In addition, the cortical endoplasmic reticulum of adjacent plant cells is interconnected by cytoplasmic strands called plasmodesmata that traverse the common walls and play a role in cell-to-cell communication. Okay, it sounds at least little strange because it's very deep in detail. This is biology of plants, not more, not less. So we are going to finish this paragraph. The endoplasmic reticulum is one of the main sites of lipid synthesis in plants, the other being the plastid. Oil bodies or lipid droplets arise in the endoplasmic reticulum and then are released into the cytosol. They are more or less spherical structures that impart a granular appearance to the cytoplasm of a plant cell when viewed with the light microscope. In electron micrographs, the oil bodies have an amorphous appearance. Oil bodies are widely distributed throughout the cells of the plant body, but are most abundant in fruits and seeds, en frutas y semillas. In fact, approximately 45% of the weight of sunflower, peanuts, flax, and sesame seeds is composed of oil. Aproximadamente el 45% de peso de los girasoles, los cacahuetes y las semillas de sésamo se componen de aceite. The oil provides energy and a source of carbon to the developing sedum. Claro, ese aceite les da la energía para esas semillas que tienen que crecer, ¿eh? En potencia tienen ese, digamos, alimento, ese pack energético fuerte. Oil bodies often are described as organelles, but this is incorrect because they are not surrounded by a membrane. Okay. O sea, incorrectamente se les llama organelles, pero es incorrecto, porque no están recubiertos de una membrana. And now we are going to continue with the Golgi apparatus. So, what is that? Golgi apparatus. The term Golgi apparatus or Golgi complex is used to refer collectively to all of the Golgi bodies, also called Golgi stacks or dictyosomes of a cell. Okay, Golgi bodies consist of 5 to 8 stacks of flattened disc-shaped sacs or cisternae, which often are branched into a complex series of tubules at their margins. Unlike the centralized Golgi of mammalian cells, the Golgi apparatus of plant cells consists of many separate stacks that remain active during mitosis and cytokinesis. 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 Let's check this. It's once in a life, this word. I'm totally sure that I'm not going to use this word anymore, but who knows? Who knows? Cytokinesis. Cytokinesis.
Cytokinesis.
Cytokinesis.
Cytokinesis. Cytokinesis. Cytokinesis.
Cytokinesis. Oh, cytokinesis.
Cytokinesis.
Cytokinesis. Cytokinesis is just the other way around. It's very different from writing form. Cytokinesis. Cytokinesis. Okay. So during mitosis and cytokinesis.
Cytokinesis.
You see? Cytokinesis. Cytokinesis. The Golgi apparatus is a dynamic, highly polarized membrane system. Customarily, the two opposite poles of a Golgi stack are referred to as forming and maturing. Forming, or CIS, and maturing, or trans phases. The slacks between the two phases comprise the middle or medial cisternae. An additional structurally and biochemically distinct compartment, the trans-Golgi network, occurs on the maturing phase of the Golgi body. Golgi bodies are involved in secretion. In plants, most of the Golgi bodies are involved in the synthesis and secretion of non-cellulosic polysaccharides, hemicellulose, and pectins, destined for incorporation into the cell wall. Evidence indicates that different steps in the synthesis of these polysaccharides occur sequentially in different cisternae of the Golgi body. Golgi bodies also process and secrete. Let's check this again because I check already, but I just forgot. Secrete. Okay. Golgi bodies also process and secrete glycoproteins that are transferred to them from the rough endoplasmic reticulum via transition vesicles. Vesicles es bicicleta, no puede ser. Y vesículas, vesicles. Let's check. Vesicles. Okay, was okay. These transition vesicles flow from the endoplasmic reticulum to the forming phase of the Golgi body. The glycoproteins are transported stepwise across the stack to the maturing phase by means of subtle vesicles. There are then short in the trans-Golgi network for delivery to the vacuole or for secretion of the cell surface. Polysaccharides destined for secretion of the cell surface may also be short in the trans-Golgi network. A given Golgi body can process polysaccharides and glycoproteins simultaneously. Newly formed vacuolar proteins are packaged at the trans-Golgi network into coated vesicles, so called because these vesicles are coated with several proteins, including clartrin, with which forms cages around the vesicles. Glycoproteins and complex polysaccharides
Destined.
destined. So simple. Was just a stress in at the beginning. Destined.
Destinada.
Oh yes.
Destined.
Destined. So it's marvelous. Destined. Okay, so glycoproteins and complex polysaccharides destined for secretion at the cell surface are packaged in non-coated or smooth-surfaced vesicles. The movement of these vesicles from the trans-Golgi network to the plasma membrane appears to depend on the presence of actin filaments. When the vesicles reach the plasma membrane, they fuse with it and discharge their contents into the cell wall. In enlarging cells, the vesicle membranes become incorporated into the plasma membrane, contributing to its growth. The secretion of substances from cells in vesicles is called exocytosis, exocytosis. Mm, ay, really, really new for me.
Exocytosis.
Exocytosis. It's like a robot. Exocytosis. So, okay. The secretion of substances from cells in vesicles is called exocytosis. Substances are also transferred from the cell wall to the trans-Golgi network or vacuoles through the formation of vesicles called primary endocytic vesicles or endosomes. At the plasma membrane, the uptake of extracellular substances by unfolding of the plasma membrane and pinching off of a vesicle is called endocytosis. I can imagine that is the same. Exocytosis is endocytosis. Yes, for sure. I don't need to check this time. So we are in the last part of this paragraph dedicated to the Golgi apparatus. So the endoplasmic reticulum and Golgi apparatus are components of the endomembrane system. Thus far, we have considered the various components of the protoplast in isolation. With the exception of mitochondrial, plastid, and peroxisomal membranes, however, all cellular membranes, including plasma membrane, nuclear envelope, endoplasmic reticulum, Golgi apparatus, tonoplast, and various kinds of vesicles, constitute a continuous interconnected system known as the endomembrane system. The endoplasmic reticulum is the initial source of membranes. Transition vesicles from the endoplasmic reticulum transport new membrane material to the Golgi apparatus, and secretory vesicles derived from the trans-Golgi network contribute to the plasma membrane. Whoa, such a long sentence. Oh my God. The Trans-Golgi network also supplies vesicles that fuse with the tonoplast and thus contribute to formation of the vacuoles. The endoplasmic reticulum, Golgi apparatus, and trans-Golgi network therefore may be considered a functional unit in which the Golgi bodies serve as the main vehicles for the transformation of endoplasmic reticulum-like membranes into plasma membrane-like and tonoplast-like membranes. Okay, so it was a little tough today. I'm sorry because the vocabulary was brand new for me, at least. It's very, very, really specific. But of course, you can imagine that if you are talking about Golgi apparatus or you are talking about the endoplasmic reticulum, it's not going to be so easy. So, friends, I hope you are going to pass a good day, a good weekend. I hope you the best and see you very soon in only 1 week. Please remember to subscribe to the channel Estrategias Diarias Cultura, and Estrategias Diarias Ingeniería, and a brand new channel that is called Estrategias Diarias Gold. Yes, Gold. The idea with this new channel is to summarize the best of the three worlds. Estrategias diarias cultura, and estrategias diarias ingeniería. All together, the best is selected and put into the new channel. Maybe in the future, it's going to remain only one channel in order to do it better. But who knows? For the moment, we are going to have 4 channels. But please subscribe to the number gold, the gold one. The gold is going to be, I think, is going to be the most important of the 3. Who knows? Let's see. Help to get. Okay, have a nice day and see you very soon with new videos for you. Goodbye. Bye-bye.